get things going. Oh, excuse me. Yeah, so what questions do people have on material, course stuff, anything like that that would be helpful to work through this evening for a review session before the exam?
I was wondering if you could maybe kind of go over signal detection theory again.
Yeah, yeah, absolutely. Thank you, Lauren. I'm gonna go through uh and we will open up perception vision. Okay. Um, all right, can you all see these slides here?
Yeah, we can.
Okay, perfect. Um, so working through here, signal detection theory. Um, so again, Lauren, what we're looking at here is just factors that are going to go through and impact absolute threshold. Okay. Remember that if we're talking about absolute threshold, we're talking about so the bare minimum amount of something in order for a person to detect it about 50% of the time. Okay. So if this is, let's say, we talked about in class, like a hearing test, that's pretty easy. If I go through, throw on headphones like this, right? And you're going through and beeping in one ear or the other, how many decibels does it have to be? Like, what's the lowest number of decibels for me to detect something at least half the time? And that would be the absolute threshold for hearing, right? Same thing if you put me in some goggles or something like that. What's the faintest amount of light that you could, the dimmest amount of light that you could sort of flash in front of my eyes, and I could sort of register that something was there, right? So this is that absolute threshold. Now, signal detection theory involves sort of different types of things that impact sort of what that level is, right? There are certain things that can go through and increase it, decrease it, and that's kind of the idea behind signal detection theory. Does that help kind of answer or clarify some of those questions?
Yeah, that does. Thank you.
Okay, cool. Other stuff that people ran into.
I wanted some clarification on like dopamine, melatonin, and cortisol, like stuff like that, and like what their main like function is for like sleep and stuff.
Yeah. So is that are we from the sleep lecture?
Yeah, it should be.
Okay. So
Mostly dopamine and melatonin, I think.
dopamine now is that from the book?
It might have been. I just
Yeah.
remember. Yeah.
Okay. Yeah, in terms of good question here, so we won't sort of pop with that open. Good question about specific neurotransmitters. Honestly, um, it's really complex. So you've got a lot of different neurotransmitters playing roles in sort of this. Melatonin is one thing that came up, and so that's going to be something that as your body's producing that sort of instigates sleep. So the other things are coming through in complex reactions. They do kind of different stuff. You don't need to worry too much about that. Just kind of know, hey, these are things that are kind of in kind of linked to but I'm not going to sort of ask you to tease apart the difference between like histamine and dopamine in terms of their specific actions. We just didn't get that far into things. So
Okay, thank you. I appreciate it.
yeah, no problem. Good question. Other questions?
I actually have another question.
Yeah. No, go for it.
um,
Yeah,
could
absolutely.
you define like the functions of the basal ganglia?
Yeah, functions of basal ganglia. So for our purposes, let's go through brain structure. Show this. Okay. Basal ganglia, right? This is going to be one of those subcortical structures. Again, if you go through and you pop the top of the cortex off, right? So you're looking underneath the cortex at the brain. We're still at the forebrain, but there are some of these subcortical structures. Basal ganglia, the thing that you should be thinking about in terms of basal ganglia, think about that primarily as involving coordinated movement. Okay, so my ability to go through and move my hands smoothly through air like this, right, is in part sort of due to functions in the basal ganglia. Okay, a lot of dopamine tracks running through your basal ganglia involved in movement, things along those lines. Again, as we talked in class, kind of the issue there when people develop Parkinson's, right? What we see is a deterioration of some of the neurotracts in there, and we see deficiencies in sort of dopamine action in some of those dopamine neurons, and so that's where you get some of the tremors and the physical impacts of Parkinson's disorder, and a lot of that is due to impairment within the basal ganglia and sort of structures in those regions. And so, so long as you sort of understand basal ganglia is kind of related to, I mean, it does other stuff, but kind of for our purposes, we can think about movement stuff as your main thing there.
So when comparing that to like the cerebellum, what
Yeah,
is like the main difference between those two?
sure. So, oh, whoops, here we go through. Get rid of that. So going back to your cerebellum, okay? Cerebellum is going to be involved more in, so the thing about your basal ganglia and kind of smooth movements. And so like I'm just sitting right here and sort of my ability to go through again, if you look at or see people, Michael J. Fox is one of the prominent celebrities that struggles with Parkinson's. You see a lot of tremors and a lot of difficulties with smooth movement types of things, right? That's going to be basal ganglia. Cerebellum, this is going to be more of that in terms of movement and coordination, kind of that vestibular sense. Okay. In terms of thinking about the cerebellum being more involved in terms of things like balance and coordination types of things. So I can I'll have tremors and shakes if I have deficits or sort of damage to the basal ganglia tracts. Cerebellum's a different thing. Like you would expect difficulties with balance, standing, sort of coordination of sort of stuff and movements and things along those lines, as opposed to the fine-tuned smooth movements given deficits in basal ganglia. So yeah, a good sort of way to try and sort of tease those apart, but that's kind of what we were thinking about with that.
Okay, so basal ganglia is smooth movement, and then cerebellum would be more balance and coordination, correct?
Yeah.
Okay.
So that's a good way, at least for our purposes. And again, with all of this stuff, if you were to get into like an intense neuro class, you start to get into more of the fine grained stuff. But I think on a broad level for our purposes, that's a fine way to go through and differentiate general functions of those two regions.
Okay, perfect. Thank you.
All good questions. Okay. Couple of questions. What would you prioritize while studying? I'd prioritize everything. That's not maybe an answer that's you might want, but like again, anything is fair game, right? So want to make sure that you got a good handle on sort of all of it. I wouldn't say necessarily prioritize sort of one thing or the other. Honestly, if I was going to go through at this stage of the game and do some stuff, working through carefully the book chapters, right? Because there's just going to be so much. If you've gone through and looked at the book, there's so much overlap with the stuff we've already covered in lecture. So if you have good lecture notes and you haven't gone through and actually read the book chapters, taking notes and stuff like that, I would for sure spend some time doing that. One, it's going to reinforce the stuff that we've covered in lecture. And then two, remember that the questions 31 through 40 are going to just be from the text, right? And so if you haven't read through the book, you're going to be probably pretty lost on a quarter of the quiz. So that's what I would say in terms of, I would not prioritize any one thing. I would say sort of make sure that you go through and cover it all. But if at this point you haven't gone through and looked at the book, I would strongly encourage people to go through and make sure that they got that under control. Okay. Looks like Bell is asking, could you go over the charts of the sections of the brain as well as brief descriptions of their purposes? Absolutely. So again, remember that with this, we're going to go through and I'm going to give you a diagram of something, right? So we just want to make sure that you know kind of just different areas and things that are going to be. Whoops. Sorry about that, folks. Can everybody still hear me? My connection cut off. You will hear me all right?
I can hear you.
Okay, cool. All right, going through and talk about brain structures. So again, wanting to think about brain structures as we can section these off into forebrain, midbrain, and hindbrain. Again, sort of think about this as an evolutionary structure, sort of moving backwards with things. For your cortex, right? Again, we're thinking about sectioning off different cortices. Again, with your, oops, there we go. Thinking about, you've got two hemispheres, right? You've got the right hemisphere, the left hemisphere, relatively sort of connected to one another, or excuse me, sort of parallel to one another, symmetrical there. And on each side, you have different sort of regions or cortices or lobes within your cortex here. So you've got your frontal lobes, that's going to be right here, your parietal lobes on top, your occipital in the back, and then your temporals on the side. Okay, your frontal lobe, this is going to be primarily involved in like a lot of planning, attention, you know, there's speech stuff going on there, some movement. But again, we think about your frontal lobes primarily is thinking about that in terms of executive functioning types of things, okay? Your ability to sustain tasks, direct attention, activities kind of like that, right? And then again, obviously on the left side and the left frontal, we've got some speech centers and things, but generally, thinking about that, a big piece of that frontal that makes us human is the ability to sort of inhibit actions, plan ahead, sort of take sort of measured steps on things. That's what's going on a lot in the frontal lobe. Temporal lobe, again, the left side, a lot of speech going on here, smell, hearing, things like that, predominantly going on in your temporal lobes. Parietal, up here, we're seeing some sensory information. Again, that band on the front of your parietal, your somatosensory cortex, that's sort of pulling in information about sort of what's going on, what you feel in the world, sensations and things like that. You got some taste, some speech stuff going on back there, and then your occipital lobes, primarily think about those in terms of vision. Okay. We talked about your somatosensory cortex. Okay, that's these strips that are right on the front of your parietal. Your primary motor cortex, that's controlling sort of hands and things like that. Remember, oh, hey, but my left cortex is controlling my right side of my body. My right cortex here is controlling the left side of my body, and that being on just the back side of your frontal. Your prefrontal cortex up here, that's the part that's really focused on regulation, attention, things along those lines. You've got your auditory cortex, and that's going to be primarily on the left side here, your primary visual back there in the occipital. We talked about aphasias. Okay, limbic system. Okay, hey, hop down. Your limbic system. This is going to be a number of structures, subcortical forebrain structures that are going to be involved in like memory, emotion, sort of things along those lines. Okay, so your hippocampus. That's these kind of hippocampus means like seahorse in Latin, I believe. This is going to be associated with a lot of memory function. Okay, the storage and sort of processing of long-term memory is going to be involved. Your hippocampus, your hippocampi are going to be involved in that. Your amygdala, right here, these little kind of eight almond-looking things on the structures on the side. Those amygdala are involved in a lot of, or have been linked to some emotion processes. Highly fear, sort of anxiety, sort of types of things, some anger stuff going on. But we think about amygdala is associated with some emotional processing. Your thalamus, this is just kind of a relay station. This comes up, things come into your thalamus, get sent out to other places, come from your brain into your thalamus out to other places. Just kind of a switch yard there in your brain in terms of directing traffic. And then your hypothalamus. This is a structure that starts to become involved in sort of basic sort of functions for life. So a lot of things related to sleep, feeding, fight or flight, stuff like that, mating behaviors and things along those lines, reproduction drives and stuff like that, sort of in being involved in your hypothalamus. This is a structure that's going through and impacting things like your pituitary and other glands within your endocrine system. Okay. Also here, basal ganglia, we talked about the basal ganglia, what's going on there. Midbrain, right? This is just this kind of sort of middle brain structure here. The only thing we need to worry about with this is the reticular formation. And here, this is just going through and acting as another relay station. Reticular formation is going to be involved in sleep, arousal, sort of things like that for the most part, some attention and movement reflexes. Again, don't worry about if there are multiple functions associated with different brain regions. Okay, that's as we talked about, like any behavior doesn't like just exist in one part of your brain. There's lots of different parts that go through and contribute to stuff. So if you got two things that say that it involves movement, then just know that it's not a big deal. I'm not going to sort of ask you to sort of tease specifics of those apart. Then your hindbrain, this is going to be your cerebellum. This is going to be your pons, your medulla. This is just like sort of basic functioning types of things. We talked about the cerebellum, what that's doing. The pons, another relay station, again, sort of involving sleep, arousal types of things. And then your medulla is going to be your brainstem. This is like just vegetative functioning. So make sure that you're breathing, that your heart's beating, that you're digesting food, all that stuff that you don't have to think about. It's just kind of going on in the background. Okay. So that's just kind of a quick run through of different brain regions. Yeah. I think, who is that? Belle, does that help?
Yes, it does.
Oh.
What's your cat's name?
This is Lion O.
Aw,
So come here, bud. Yeah,
that's so cute.
he's a pest. So yeah, that's not necessary. But
Oh,
he enjoys attention. So
that's so sweet.
thank you. You enjoy attention, bud? Yeah.
Oh, such a little pudgy face.
Yeah. We got similar beards. Yeah. Oh, hey, what are you doing? Okay. All right. What questions do you have? Other stuff that people have that we can help out and get clarity on.
Is there a way I could have you do a couple comprehension check problems from chapter 5?
Yeah. What was that, Tyson? Was that you?
Yeah, it was.
Okay, yeah. I don't know. I could go through and pull it up. But can you just read me the question? I'll see if I can help you out.
Yeah, let me just pull it up real quick.
I'm working too on my end to see if I can get there.
Can you do the difference between top down and then bottom up?
Sure. And we talked about this, and this is something we can go through and here, I'll just pull up slides on perception. All right. Top down, bottom up processing. People will struggle with this sometimes. The main thing to be thinking about here is bottom up processing is going to be like just the bare bones, like the kind of like think about bottom up as the who, what, when, where, like types of things, right? So if I'm looking at these figures up in my upper corner here, I look at this and I see this as a green circle, right? I see this as a green circle, right? Here's the boundary. It has a black border with a green fill. Here's the edge. I can trace my way around that, right? That's just bottom up stuff. Same thing here is a vertical bar. Here is the height and the width going down. Here's the edge. Here's the foreground, the background. It's just like, here's the basic stuff, okay? Top-down processing is different. This is what's going on. This is higher order mental processes that are going through and starting to impact your perception of things. Okay. And this is why I think that Brain Games clip really drives home that what we're experiencing out there, and we experience like, oh, I saw it with my own eyes, right? Like, or I heard it, and sort of like it's real if I saw it or heard it, right? But anything that our brain is processing is just some like story of what's going on out there. And it's a story that's generally useful for us, that makes some sense, but it doesn't necessarily correspond to what's actually sort of out there in terms of the real world, right? So again, example of this is perceptual set, right? We looked at this figure, and we can look at this, and if you get it, right, you can look at this and identify this as a whole as like an old woman with her head kind of pointed down, or you can look at this as a young woman with her head turned slightly away. And you can switch back and forth, right? Now, there's probably one that's easier for you to see just because that's kind of what your brain's making the most sense, but we can go through and switch it so you can see either one of these, right? And this is a shift in perceptual set. You're seeing a completely different image based on how you're sort of setting your perception, right? Now, if you ask me either way to go through and trace the lines and identify sort of the black from the white and where things are at and the borders and lines, I can do that. That's the bottom up stuff. It's the top down that's changing my experience of what that is. Okay. Same thing we talked about this image here. Bottom up, I can go through and identify that this is a curved line, this is where it is, right? I can go through and identify that in terms of bottom up. But to make sense of this, there's some top down stuff going on, and my brain sees this. And in the text, they have a nice section on Gestalt psychology. And sort of the idea behind Gestalt is the whole, right? And the way our brain will go through and start to fill in information in order for us to sort of see things that we might expect or make sense to us, right? Now, Tyson, when you look at this image, what's the first thing that you see? You see a mouse or a face?
I see a mouse.
You see a mouse. I tend to see a mouse as well, right? And so I go through and look. Now, is it a full drawing of a mouse with all the lines connected? No, but my brain sort of has gone through and says, okay, this is something that I recognize as a mouse, right? But if I were to go through and proceed this image by a similar line drawing of a face, a line drawing of a face, a line drawing of a face, and a line drawing of a face, my brain is likely not to, if this pops up, I'm likely not to see it as a mouse. I'm likely to see it as a face, right? Because that's what I've been seeing and detecting and processing. Much more likely to say this as kind of an old man with glasses and kind of a pointy nose and sort of a round head, right? But if I go through and do the same thing and proceed this one after the other with this, and then I show you this, and then I show you this, and I show you this, then the mouse is more likely to pop up, right? And so again, the image is exactly the same. There's no difference across these two. It's just how my brain is going through and processing it based on expectancy. Okay. And again, in the book, that Gestalt chapter talks about different things like continuity and sort of consistency in terms of shapes. The brain games demonstration had a cool thing where you got through and likes the tops of letters, and if you look at it, it looks like you can read it perfectly. But then if you pull the bottom off of it, it's something completely different, right? Just showing how our brain, from a top-down way, is taking incomplete information and sort of turning it into something that makes sense for us, right? And that being a very useful thing in general, but sometimes it can be wrong. Okay, which is kind of will be a theme this year. Like our brain setup and our psychological processes are doing things for speed and efficiency and probably to keep us safe historically, but it's not always super accurate. Does that help tease some of that apart?
Yeah, that helps a bunch actually.
Okay, cool. Other questions? You feeling okay with everything?
Is there a way I could have you go through and show the eye diagram again and all their functions?
Oh yeah. There's an easy way to do that right here. Perception, there we go. Cool. So for which eye diagram are you looking at, Tyson? This one?
Yeah, that one.
Okay, so here, this is just basic structure of the eye. And again, remember, you want to go through and make sure you got diagrams down so that you understand kind of where things are at, because I'm going to pull something to test you on that. But for this one here, we've got the eye, right? And just basic anatomy. Here's your cornea. That's just the outside of your eye, okay? The lens is sort of what's focusing light, your pupil, the hole in the middle, and then your iris, that's the colored part, right? That sort of determines how much light goes in, okay? What's happening is light is coming in through your pupil, and it's getting focused through your lens and getting targeted back here on your retina. Retina is a small space on the back of your eyeball that contains your rods and your cones, okay? And in the center of that, you got this little divot, that's your fovea. Okay, that's where sort of if your lenses are working correctly and you don't need sort of contacts or thick glasses like me, what your lens is doing is it's focusing that light specifically right here on the fovea. Now your fovea is where your rods are concentrated, right? Rods are looking at kind of like, think about these as your high def receptors. They get color, they see fine detail. Anytime I'm looking directly at something, I'm seeing a very bright and sharp image in there. Again, so I don't need eye correction, or if I got glasses and things like that, that's helping of what's going on, right? Now your retina is larger, right? And so what we see sort of on the periphery of the retina, we'll see your cones, okay, or excuse me, your rods. Cones are up here, cones are capturing color and detail. Rods, this is just kind of, they're monochromatic. They're not getting color. They're just getting shades of sort of light, but they're very, very sensitive to light. Okay. And your rods are existing sort of in the highest concentration on the periphery of your retina. So again, if let's say I'm living, you know, I don't know, like hundreds of years ago, right? And I'm trying to go through and see someone send me a signal at night, so because they've got a fire and I'm trying to see it, right? Looking directly at it is probably not going to be the best way to detect something, right? If I'm going through and catching something out of my periphery, that's much more sensitive to sort of lights than the presence of light than your fovea is going to be. That's looking for detail, okay? So what's interesting, get down, is, again, if we look at this part, this is just kind of a close-up of your retina, okay? And what you see here is you see rods and cones kind of lined up. But the business end of these cells, kind of the thing that's picking up the light, is actually pointed at the back of your head. They're pointed at the back of your eye, right? So light is coming in, it's hitting sort of the surface of the retina, but then what it's doing is it's kind of going through this membrane and sort of hitting the actual back of your eye right here. And this is where that light is triggering your rods, your cones, right? These rods and cones then connect to these bipolar cells. These are just kind of connecting cells, and then to these ganglion cells. And the axons of those ganglion cells, that's what's making up your optic nerve going back into your regions of your brain. Yeah. Does that help in terms of sort of kind of the anatomy and sort of the different functions and sort of how everything's working here?
Yeah, that does.
Okay, cool. I guess going on, just Tyson, the same sort of thing. You comfortable with neural pathways, visual pathways? What's going on? This gets a little confusing in terms of right to left, left to right stuff.
Oh, I might appreciate it if you could go over it one more time.
Okay, cool. So when we're thinking about sort of visual processing, where these receptors are coming from, what they're detecting, right? If I'm looking, so right now I'm looking at sort of the camera, right? So you can see me waving my hands around, or maybe you can't because I think you're just looking at the screen, but I am in the side here. But if I'm looking at this, actually, yeah. If I'm looking at this right here, I'm just okay. Now just so you can see me. So I'm looking straight here at the camera at you all at the moment, and what's going on is everything on the right. So the retina of both of my eyes, right on the right side, the right sides of my retina on both eyes. This is picking up my left periphery. And if I looked and split my eyes down the center and the left side of my retina, this is picking up my right periphery. Okay. And what happens is that, again, if I sort of have my finger out here and pull it like right about there is where I can see if I'm looking straight ahead in my periphery, that's the boundary of my periphery. This movement that I'm detecting right here is going directly in my visual cortex on my left side. If I pull out my or my left periphery, there I can kind of see something that starts to get in. That's strictly going, it's being captured by the right sides of my retina, so it's catching that. And that's going into my left side. Okay, so we got a crisscross going on. Anything that's going on here is being uniquely processed on the edge on my left side, and everything sort of happening in my left is being uniquely processed in my right side. Now, the things happening here, right, the center that I'm looking at, that's being captured by both sides of my brain or both sides of my retinas. And so that's going to both hemispheres simultaneously. Okay. And so what we start to see if we jump back into that split brain procedure, if I have my corpus callosum, right, the bundle of nerves that connects my two hemispheres, if that's severed, you would think that would really screw people up, but it actually doesn't. Like, people function just fine with that, right? Particularly for sort of vision, because generally when I'm looking around, if I'm reading a book, things along those lines, everything that I'm sort of focusing on is going to both my hemispheres simultaneously, okay? But if I go down through and I have a situation here where I'm looking at a fixation point, I'm just flashing something into my right periphery, that's going uniquely into my left hemisphere. If I go something into my left periphery, that's uniquely going into my right hemisphere. And this is where we saw that split brain procedure thing going on, where the patient in the video was able to say anything that was flashed in the right periphery, because that's going into my left hemisphere. I mean, the image is going into my left occipital lobe, and then that's being transferred over to other regions in my right or in my left hemisphere. I've got language predominantly over here, so I'm able to go through and describe it, right? But anything going on in my left periphery, right? That's going into my right brain. My right brain's not so good with language, right? But it's good with spatial stuff, right? So if something flashes into my left periphery, which means it's going into my right hemisphere, I'm not able to say what I saw. I don't have words to say. I can't describe what I've seen, right? But if I close my eyes and with my left hand, I can go through and draw it, right? And so what we saw in that video was really interesting. So if you flash an image into both the right and left periphery, okay, I'm able to say whatever I saw in the right periphery, but I'm able to draw with my left hand what happened in the left, or what was shown in the left periphery. Okay. So some interesting sort of crisscross stuff going on with that. Does that Tyson help kind of sort through that piece of things?
Yeah, it does.
Okay, cool. Good questions. Yeah, that's probably one of the trickier sort of concepts in sort of keeping that straight. So other questions that people have.
I have one last question about chapter 6.
Mhm.
How in depth do we need to like understand that material? Just because it was like kind of fairly recent and our last chapter. Just how much, like how familiar should we be with that?
Aubrey, do you mean chapter 6 or chapter 5?
Whatever the last chapter. What is chapter the one we went over on Monday yesterday?
Do you mean the book chapter or the lecture materials?
The lecture material. It dealt
Oh,
with
okay.
like the sense like of smell.
Yeah, your non-visual stuff. Yeah,
Yes.
yeah. Generally know kind of what's going on with sort of those non-visual senses, and kind of know what I would know is what the signal, what's being detected by those symptoms, how those things are being transduced into electrical activity that we then go through and do, or that we then sort of our brain is able to work with and process. So yeah, I would sort of still have the same sort of amount of knowledge of that as everything else.
Okay, perfect. Thank you.
Categories. Sierra, I saw that you had asked about going through the ear and hearing. Yeah, that's another one that ends up being a little bit more involved. Let's see, non-visual senses. There we go. All right. Okay. So for the ear, okay, basic structures, the stuff that you can see on the outside, right? It's just your pinna and your ear canals, right? So this is what's going on. Your ears out here capturing sound as it's coming through the air. Your ears, right? So your outside of your ears, your pinna is capturing that. You got like little creases and serrations there, and then that's sort of going through, focusing those sound waves down into your ear canal. Okay, those sound waves are going into your ear canal, vibrating your eardrum. Okay, and the vibrations on those eardrums goes through and it shakes and vibrates the small bones in your ear, the stirrup, the hammer, the anvil, right? This is stuff that we learned in elementary school. I'm like, okay, good enough. But where things start to get complicated is when it sort of goes into the cochlea. Okay. Now, the thing, in order to understand what's happening in the cochlea, have to know exactly what's going on with the waveform that's coming through the air, what we perceive as sound. Okay. Sound isn't real, right? Sound is just a physical wave going through the air that our brains have gone through and taken sort of that and translated into an experience that help us go through and detect things and things along those lines, right? So sound is a thing that's completely made up by our brain, right? Sound out there in the universe, absent ears, isn't a thing. It's just sort of waves moving through air. But what we've developed in our brain is a way to go through and take that or that physical energy, translate it into something that is useful for us. And sort of this is what we call our audition, right? Our sense of sound. And the stimuli that we're picking up are these sound waves, okay? And a couple of different features, these sound waves start to get translated into different auditory experiences. The pitch, this is how fast that sound wave is moving, okay? Something that's very slow wave like this one, okay, that's going to be a lower tone. Something that cycles much more quickly, right? It's really fast moving up and down. That's going to be a higher pitch, okay? And again, I talked in Mickey Mouse voice and in low voice in lectures. We won't do that for the recording, but you kind of get the idea. Like this is your bass and this is your treble, right? The bass is slow, right? Sort of going through, and those waves carry farther, right? Your treble, your sort of higher pitch, that's moving. Those are waves that are moving much faster, but they carry much less far through sort of media and things along those lines. Okay. So again, the frequency and the amplitude is what we're focusing on here. Now, what happens when whatever sound is coming in, it vibrates our eardrum, which then vibrates the small bones in our ear, which then correspond to vibrations in this cochlear window. Okay. And if we were to look at your cochlea is this snail-looking organ right here. If we were to go through and unfold that, roll it out like a sock, right? What we would see is inside of that, we have what we call the basilar membrane, okay? And this is just kind of a flat sort of piece of, well, if you go through and take a look what we see, if we do a cross-section of this, you've got fluid in the bottom chamber, fluid in the top chamber that goes through and runs through the whole thing, okay? But when we get vibrations at the window, what it does is it starts to vibrate the fluid inside your cochlea, okay? And you've got your basilar membrane in here that's just kind of like a tongue just kind of hanging out, flapping around in there, right? Now on the top, you've got kind of a sturdy structure, and this is where inside your basilar membrane, you got these little hair cells. And the tops of these hair cells are attached to this rigid top structure. The bottom, though, is all kind of loose. So if the fluid beneath it gets loose, it goes through and shakes those hair cells, okay? And this is what stimulates it. It gets turned into electrical activity that's picked up and transmitted through the axons of your hair cells into your auditory nerves back into through your thalamus and into your auditory cortices and your temporal lobe, okay? Now, how we go through and experience different sort of characteristics of noise depends on a couple of different things, okay? Volume, right? How loud something is. This is just a function of the number of hair cells activated. And you can start to see this. If I go through and I have something that's very soft, right? You've got someone who's whispering and you're trying to go through and hear what's going on. The vibrations coming through the air are very low amplitude, right? They're not big, they're not strong. And so you're getting a vibration at the cochlear window, but it's not very much, right? And so if we think about the consequences for the disruption of fluid in your cochlea, it's not moving that around a whole lot. So this basilar membrane isn't going to move all that much. And so you're not going to get many of these hair cells activated, and it's going to be a very soft sound, okay? Now, if someone starts shouting at you, or sort of a truck drives by, or sort of a train or a firecracker or something like that, that's a loud noise. The amplitude of that sound wave is very huge. It's very, very disruptive, okay? Think about soft sounds as throwing a pebble in a lake. Loud sounds like throwing a boulder in the lake. It's much more disruptive. And as that goes through and you get a much stronger vibration at your cochlear window, what it's going to do is it's starting to move that basilar membrane around a whole lot more, and that's going to start activating these hair cells all up and down this, right? And so the number of hair cells being activated, that's sort of the physical property that our brain translates into the experience of volume, okay? Now, pitch is a little bit different. Remember, pitch is the frequency of that wave. If it's a slow wave, it's a low sound. If it's a fast wave, it's a high sound, okay? So one way that our brain codes pitch is the frequency at which these hair cells are firing. If I've got a really high-pitched sound coming in, it's going to make a really fast vibration at that window. And so the fluid is going to be shaking really fast. And so it's going to shake these hair cells really fast. And so the frequency of those hair cells shaking is in part going to code pitch, right? If they're very slow moving around, it's going to be a low sound. If it's very fast, that's going to be we're going to perceive that as a higher pitched sound. Okay. Now, also in addition to that, remember I talked about these waveforms, your bass, your slow wave stuff, that's going to carry out a lot farther. It's going to reach farther out in distance than something that's very sort of high frequency and fast. And so the other thing that's getting coded as pitch in your cochlear organ is where on this basilar membrane things are being activated. Okay, so high pitched noises, they're going to sort of start and sort of disrupt and start to make sort of vibrations sort of kind of here at the base, but it's just not a very far moving wave. And so it's not going to sort of probably make it all the way to the very tip. But if you've got a low sound, someone rolls by, they got their bass cranked up, very, very low frequency sound, that's powerful. That's going to carry a long ways, and it's going to go through and start to create slow vibrations all the way down to the very tip of this, right? And so if we get activation in the cells here all the way at the end, that's going to be encoded as a low frequency or a low tone. Okay, so volume being coded just by the number of cells that are activated. If you got something really large, you're standing by heavy equipment, it's going to be really hard, or it's going to go through and shake this a whole bunch. Everything's getting jostled around. That's going to be experienced as a loud noise. Something that's very soft, it's not going to shake things very much. You're just going to maybe get a little ripple here. It's not going to activate a ton of hair cells, so that's going to be soft. Okay, pitch. Determined by the frequency, how fast this is moving, right? And then at what point along the basilar membrane that's being activated. The low stuff happening here at the end, high stuff happening here at the base. Sarah, does that help in terms of sort of understanding kind of mechanisms and what's happening with this? Thumbs up, feeling good about this? Okay, cool. All right, yeah, those so that piece is mechanisms of hearing. And Aubrey, to your question, yeah, wanting you to know stuff about this. Hearing is pretty complex in terms of there's just a lot of different things going on. Olfaction, pretty straightforward, right? It's just what we're picking up is just different chemicals in the air, right? These odorant chemicals. And sort of these odorant chemicals go through and bind to receptors, right? We have about 350 unique olfactory receptors, but we can smell more than 350 things, right? So what's going on with that is combinations of different odorant molecules that are going through and activating things, right? So if I smell, say, pizza or something along those lines, right? There's a lot of different things that are coming through that is sort of capturing all these different smells. So I'm sort of smelling stuff that's more complex, right? So you've got all possible combinations of 350 unique receptors that are binding to different odorant molecules, so that provides us with, and other animals have better senses of smells, right? But ours is, I guess, fine for sort of what we need them for. And so we can still sort of go through and identify a lot of unique stuff, right? Taste, again, the same thing. Sort of it's a chemical sense. We go through, something goes into our mouth, sort of our saliva carries it down in between the pores of these papillae, down into where our taste receptors are, right? We've got our taste bud is actually a collection of these taste cells, right? That's going through and picking up stuff that sort of our saliva is carried down in between the crevices of our tongue. And this is where we're going through and identifying different flavors related to sweet, sour, bitter, salty, umami, things like that. And that's going through our to our gustatory cortex. Okay, cutaneous sense. This is a physical sense, right? So in your skin, right, what we do is we have receptors that are unique to different types of things going on or that we're experiencing, right? We've got pressure sensors, right, that detect changes in pressure. So I can tell if I'm touching things because it starts to deform my skin, right? If I put my hand down, the receptors in the skin are changing shape. And so I'm perceiving that as sensation of pressure and touch, vibration. If I go through and sort of my head's pretty smooth, but it's got some tiny hairs on there, right? What it's doing is causing some smooth and vibration. Those vibrations are being picked up as texture, right? You think about you rub your hand over a piece of burlap or something like that, right? It's going to be rough, or sandpaper. I experience that as rough because what's happening is I move my hand across it. It's creating vibrations in those receptor cells that my brain is then transmitting that vibration as an experience of texture. Okay. Then we have sensors that are specific to heat, sensors that are specific to cold, sensors that are specific to tissue damage, and sort of thinking about different types of pain that we can experience, right? Intense pressure, if something squeezes something really hard, it hurts, right? This isn't like sort of just basic pressure touch. This is, oh, it got hard to a point where it's starting to hurt, could damage my body. And so my brain is registering that as pain. Things like acids, heat, capsaicin, things like that, right? If I go through and sort of take habanero sauce and sort of rub it on the inside of my nose, that's going to hurt. Even if I get some on my skin, right, so it can start to burn. We've got special receptors for that. And then when we have things like disruption of blood, muscle damage, things along those lines, spraining an ankle, or sort of something along those lines, we've got different receptors that are picking that up and reading that as pain, right? And so that's what's going on. I realized I wasn't sharing my screen for any of that, but as you're working through, if you're following through on the non-visual senses slide, you just kind of see working through those different senses and sort of understanding just kind of the basic mechanisms of that. Was that helpful, Aubrey?
Yes, it was. Thank you.
Yeah, cool. All good questions, folks. Other questions? Feeling good? Okay, cool. Well, thank you all for jumping on. So we'll go through and make sure that we send this out for people who might not have been able to pick up. But thank you for linking in. If you have any other questions as you're going through getting prepped, just let me know. Otherwise, we'll catch you all in the morning, and yeah, get some rest. All right,
Thank
folks.
you.
Yeah, we'll see ya.